Hi everyone, in this video we are going to do a short recap of what you previously learned during your anatomy and physiology course last year, especially pertaining to neurons and neurotransmission. The nervous system is divided into two main components, the central nervous system, which consists of the brain and spinal cord, and the peripheral nervous system, which consists of the nerves and ganglia outside of the brain and spinal cord. Nerve tissue is the main tissue component of the nervous system and is primarily composed of neurons and supporting glial cells. Neurons are basic nerve cells which transmit messages throughout the nervous system, resulting in functions as diverse as tasting, thinking, and moving. Glial cells, on the other hand, provide structural and functional support to neurons. There are two main types of glial cells. You have the microglia, which provides phagocytic role in terms of destroying invading microorganisms, removing cell debris, and promoting tissue repair. And then you also have the macroglia, which includes examples such as oligodendrocytes, Schwann cells, astrocytes, and ependymal cells, all of which have a variety of supportive function within the nervous system. Neurons are polarized signal transmitting cells that makes up the central and peripheral nervous system. Let's take a closer look at the main components of a neuron. The cell body contains the cellular machinery that keeps the neuron alive, for example, the nucleus. The nucleus is critical for neurons' vitality as it contains the genetic materials, or rather the genes, needed for cell division, development, and protein synthesis. Most neurons have a single axon. An axon transmits the signal generated by the neuron, or rather the action potential, through the nervous system. Axon terminals are the regions at the end of an axon that release neurotransmitters. Myelin sheath is a whitish, fatty layer that wraps around the axons of most neurons and serves to increase the transmission speed of an action potential along the axons. Axons with a myelin sheath are known as myelinated axons. Dendrites receive information from other neurons. Each neuron typically has more than one dendrite. Neurons may be classified based on the number of neuronal processes, or rather neurites, that project from the cell body or the soma. There is the unipolar neuron, in which a single axon projects from the soma, and there are dendrites that may branch off from the axon. The second type of neuron is the pseudo-unipolar neuron, in which there is a single neurite projects from the soma, but then it branches into one axon and one dendrite. There's also the bipolar neuron, in which there is one axon and one dendrite that projects from the soma. And last but not least, there is the multipolar neuron, in which there is one axon and multiple dendrites that project from the same soma. Let us now take a look at the glial cells and their various functions. Astrocytes are star-shaped cells that maintain a neuron's working environment. They do this by controlling the levels of neurotransmitter around synapses, controlling the concentrations of important ions like potassium, and providing metabolic support. But astrocytes don't just maintain the environment around synapse. An active area of research addresses how astrocytes modulate how neurons actually communicate. Because astrocytes have the ability to sense neurotransmitter levels in synapses and can respond by releasing molecules that directly influence neuronal activity, astrocytes are increasingly seen as an important factor for modifying synapses. Oligodendrocytes provide supports to axons of neurons in the central nervous system, particularly those that travel long distances within the brain. They produce a fatty substance called myelin, which is wrapped around axons as a layer of insulation. Similar in function to insulation layers around power cables, the myelin sheath allows electrical messages to travel faster and gives white matter its name. The white is actually the myelin wrapped around axons. Microglia are the brain's immune cells, serving to protect it against injury and diseases. Microglia identify when something has gone wrong and initiate a response that removes the toxic agent and clears away the dead cell. Thus, microglia are the brain's protectors. Ependymal cells in the spinal cord and ventricles of the brain are involved in creating cerebrospinal fluid. Satellite cells surround neurons in the sensory, sympathetic, and parasympathetic ganglia and help regulate the chemical environment. So this is some of the examples of glial cells, or at least the main examples of glial cells that are important in the nervous system. An action potential is generated at the origin of the axon following sufficient excitatory stimulation of the neuron. The action potential is created by movement of electrically charged particles, which is ion, in and out of the neuron through pores, which are the ion channels, in the cell membrane in a particular pattern traveling along the length of the axon. When the action potential reaches the axon terminal, it stimulates the release of chemical neurotransmitter. After the action potential has induced the release of chemical neurotransmitters, the neurotransmitters pass information to the next neuron. Neurons communicate with each other via brief electrical signals known as action potentials. An action potential is basically a sequence of changes in voltage-gated ion channels in a neuronal cell membrane that generate a nerve impulse. This upper chart is an illustration showing changes in membrane potential over time during a single action potential. So a neuronal cell membrane with a resting potential of approximately -70 millivolt depolarizes slowly until it reaches its threshold potential of approximately -50 millivolt. Action potentials will only occur if a threshold is reached. At this point, it depolarizes abruptly, which is shown here in orange. During depolarization, what happens is that voltage-gated sodium ion channels open due to an electrical stimulus. As the sodium ion rush back into the cells, their positive charge changes potential inside the cell from negative to more positive. Depolarization ends with a brief positive polarization of the membrane, after which a rapid repolarization phase begins, and this is shown in green, and the membrane potential becomes negative again. So what happens at this point is that once the cell has been depolarized, the voltage-gated sodium ion channels begins to close. The positive potential inside the cells causes voltage-gated potassium channels to open, and potassium ions now move down their electrochemical gradient out of the cells. As the potassium moves out of the cell, the membrane potential becomes more negative and starts to approach the resting potential, which is shown here. Typically, repolarization overshoots the resting membrane potential, making the membrane potential more negative. And this is known as hyperpolarization. So the membrane potential very briefly, for approximately 2 milliseconds, drops below the resting potential before returning to the baseline, which is the resting potential at approximately -70 millivolt. The occurrence of an action potential through the opening of ion channels and the resulting depolarization are local events. In order for an action potential to function as an impulse for communication, it must be directed, as in the travel of the action potential must be passed on in one direction. Stimulus conduction either happens rapidly via saltatory conduction or slowly along the non-myelinated nerve fibers via continuous conduction. Continuous conduction, which is shown in the top left corner, occurs in non-myelinated nerve fibers. So action potentials migrate along the entire cell membrane as depolarization of one section initiates depolarization of the next, and the refractory period prevents backpropagation. Saltatory conduction, which is shown on the bottom left corner, occurs in myelinated nerve fibers. So these fibers have intermittent gaps in the insulating myelin layer called nodes of Ranvier. Action potentials keep along the cell membrane as depolarization in one node initiates depolarization of the next, resulting in higher conduction velocities than possible compared through continuous conduction. On the right hand side is the conduction velocity. So conduction velocity is increased by the presence of myelination, which is what happens during saltatory conduction, and it is also increased when there is an increasing nerve fiber diameter. So as you can see here, increasing in diameter would also result in increase in conduction velocity, and myelination will also increase conduction velocity. So how do neurons talk to another neuron? Neurons communicate with one another at junctions called synapses. At a synapse, one neuron sends a message to a target neuron, which is another cell. Synapse is the point of communication between two neurons or between a neuron and a target cell like a muscle or a gland. At the synapse, the firing of an action potential in one neuron, which is the presynaptic or sending neuron, causes transmission of signal to another neuron, which is the postsynaptic or receiving neuron, making the postsynaptic neuron either more or less likely to fire its own action potential. This is a schematic description of the two main types of synaptic transmission. Chemical synapses on the left-hand side regulates the release of neurotransmitter. Most synapses are chemical, and the synapses communicate using chemical messengers, or what you know as neurotransmitter. At a chemical synapse, an action potential triggers the presynaptic neurons to release neurotransmitters. These molecules then bind onto receptors on the postsynaptic cells and make it more or less likely to fire an action potential. Other synapses are electrical synapses, as shown on the right-hand corner. And in these synapses, ions flow directly between cells. Electrical synapses operate by bidirectional passage of electrical currents through the gap junction. The current is carried by ions from the gap junctions. In typical neuronal communication in the nervous system, chemical synapses play a very important role in transmitting the signals. Electrical synapses are not important for synaptic transmission in the nervous system. However, it is by the electrical synapses that action potential are transmitted from one cell to the next in smooth and cardiac muscles. In electrical synapses, signals can be transmitted in either direction, which is bidirectional. However, chemical synapses always transmit the signals in one direction, which is very important and a desirable feature for neurotransmission in the nervous system. Let's take a look at the chemical synapse. Synapses are composed of three main parts: the presynaptic neurons, which contains the terminal buttons having neurotransmitters within the synaptic vesicles; the postsynaptic neurons, which is composed of a part of the dendrite or cell body of the neuron and contains receptor sites to receive incoming signals from presynaptic neurons; and last but not least, the synaptic cleft, which is the gap between the presynaptic and postsynaptic neurons. The presynaptic neuron uses chemical signals, or rather neurotransmitters, to increase or to excite, or decrease or to inhibit the generation of action potentials in the postsynaptic neurons. It is also possible to affect other biochemical processes, such as signaling pathway in the postsynaptic neuron. This complex interconnectivity gives rise to the rich functional communication network that is central of the nervous system. Let's then take a closer look at the process of chemical neurotransmission. The process of chemical neurotransmission is shown on the screen. Briefly, an action potential arrives at the terminal end of the presynaptic neuron, which means that the membrane potential at the terminal is altered. This altered potential causes voltage-gated ion channels to open, leading to a rapid influx of calcium. The increased calcium within the neuron terminal causes vesicles to fuse with the membrane and release neurotransmitters into the synaptic cleft. The neurotransmitters will then bind to specific receptors on the postsynaptic neuron. The effect of this binding varies, but it usually leads to altered conductance of postsynaptic neurons and the propagation of action potential. Neurotransmitters can either bind to ion channels, in which case they have a direct and immediate effect on postsynaptic function, or they can bind to G-protein coupled receptors, which results in an indirect and slower effect. Neurotransmitters can be classified based on their function. Basically, a neurotransmitter influences a neuron in one of three ways: excitatory, inhibitory, or modulatory. Excitatory neurotransmitters increase electrical excitability on the postsynaptic side through modulation of transmembrane ion flow to facilitate transmission of an action potential. Inhibitory neurotransmitter decrease electrical excitability on the postsynaptic side to prevent the propagation of an action potential. Whether a neurotransmitter is excitatory or inhibitory depends on the receptor it binds to. Additionally, there's also the neuromodulators, and neuromodulators function to alter the strength of transmission between neurons by affecting the amount of neurotransmitter that is produced and released. Neuromodulators are a bit different, as they are not restricted to the synaptic cleft between two neurons, and so they can affect large numbers of neurons at once. Neuromodulators therefore regulate populations of neurons while also operating over a slower time course than excitatory and inhibitory neurotransmitters. So what happens when the neurotransmitter binds to its receptor at the postsynaptic neuron? When a neurotransmitter binds to its receptor on a receiving cell, it causes ion channels to open or close. This can produce a localized change in the membrane potential of the receiving cells. In some cases, the change makes the target cells more likely to fire its own action potential, and in this case, the shift in membrane potential is called an excitatory postsynaptic potential, or epsb. In other cases, the change makes the target cell less likely to fire an action potential and is called an inhibitory postsynaptic potential, or ipsb. So basically, epsb, or excitatory postsynaptic potential, is any voltage change in the direction that makes a neuron more likely to fire. And epsb is depolarizing. It makes the inside of the cells more positive, bringing the membrane potential closer to its threshold for firing an action potential. Sometimes a single epsb isn't large enough to bring the neurons to threshold, but it can sum together with other epsb to trigger an action potential. ipsb, or inhibitory postsynaptic potential, is when a neurotransmitter hyperpolarizes the postsynaptic cells and makes it more negative than the resting membrane potentials, and it makes the postsynaptic cell less likely to fire. So ipsb have the opposite effect. That is, they tend to keep the membrane potential of the postsynaptic neuron below threshold for firing an action potential. ipsb are important because they can counteract or cancel out the excitatory effect of epsbs. How do epsb and ipsb interact? Basically, a postsynaptic neuron adds together or integrates all of the excitatory and inhibitory inputs it receives and decides whether to fire an action potential. The integration of postsynaptic potential that occur in different locations but at about the same time is known as spatial summation. And the integration of postsynaptic potentials that occur in the same place but at a slightly different time is called temporal summation. For instance, let's suppose that the excitatory synapses are made on two different dendrites on the same postsynaptic neuron as shown below. Neither synapse can produce an epsb quite large enough to bring the membrane potential to threshold at the axon hillock, which is the place where the action potential is triggered, which is in the box shown in blue. If both subthreshold epsb occurred at the same time, however, they could sum or they could add up to bring the membrane potential to threshold, and ultimately this would lead to generation of action potential. Shown on the screen is a number of examples of both temporal as well as spatial summation that could potentially occur. In the first graph, you can actually see that there is no summation. So basically, there is two stimuli that is separated in time. So consequently, because of the long duration of the stimuli being separated, it does not cause the epsb to add up together and thereby no action potential generation. In the second graph, this is an example of temporal summation. So what actually happens is that there is two excitatory stimuli that happen close in time, and consequently the epsb's then add up together, and this causes the membrane potential to actually reach its threshold level and consequently triggering the action potential generation as shown here. In the third graph, this is an example of spatial summation. So what happened is that there is two separate stimuli, two simultaneous stimuli at different locations. So there is one occurring at one location bringing epsb, and there's another epsb occurring at a different location. However, because it happens within sort of like the same time period, this causes the epsb's to add up together, and this causes the threshold level to be reached and consequently triggering the action potential generation as shown here on the graph. In the last graph, this is an example of spatial summation of epsb and ipsb. So at one location you have epsb coming in, and at the other location there is ipsb happening. So the changes in membrane potential of both this epsb and ipsb actually cancel each other out, and because there is no changes or there is no significant changes to cause membrane potential to reach the threshold level, thereby there is no action potential generation being triggered in this last instance. So basically this is what you have covered previously during your physiology and anatomy course last year. So it's basically an overview of what you have learned. If you need more details, kindly check out the lectures from Dr. Richard in his YouTube channel. So thank you very much, everyone. Let's move on to the next video, and that would be specifically focusing on the peripheral nervous system.